Mechanism of cryopreservation or steps which are involved in the regeneration of plants and animals involve the following steps. First of all is the selection of animal material, that what kind of material you should use for the cryopreservation. Then is different addition of cryoprotectants, freezing, storage, thawing, washing, and measurement of viability and regeneration of plants and animals. First step in cryopreservation is the selection of appropriate plant and animal material. Two important factors such as the nature and density which should be considered for the selection of plant or animal material. Any tissue could be selected for this purpose. For example, in case of plants, you can use the meristem cells, embryos and ovules, seed tissues, organs and cells, etc. So the only thing that you should be more virulent or vibrant about this is the density, that then density should be high. And second step is the addition of cryoprotectants. They are the chemicals which prevents the cryodestruction, and these are the sucrose, alcohol, glycols and some amino acids, DMSO, etc. Generally, two cryoprotectants should be used together instead of a single one, as they are more effective. Next step is the freezing. The sensitivity of cell to low temperature depends on the species. There are 4 different types of methods which could be used for freezing of the animal or plant cells. First step is the slow freezing method. The tissues or cells is slowly frozen at slow cooling rate, and the advantage of is that the cells are partially dehydrated and survive better. Second is the rapid freezing method, which involves plunging the vials in liquid nitrogen, and temperature decreases from -300 to 1000 degree rapidly and cells are preserved. Stepwise freezing method is another important method, which is the combination of both slow and rapid freezing methods, and this process is carried out in stepwise like manner. Dry freezing method. In this method, dehydrated cells and seeds are stored. Next step is storage. Maintenance of frozen cells or materials at specific temperature is very important. In general, the temperature is kept -70 to -195 degrees Celsius. Prolonged storage is done at temperature of -195 degree in liquid nitrogen. To prevent any kind of damage, continuous supply of nitrogen is done. Then thawing is usually carried out by plunging the vials into warm water bath with vigorous swirling. As thawing occurs, the vials are transferred to another bath at 0 degree. Then washing and reculturing is done. Preserved material is washed few times to remove the cryoprotectants, and then material is recultured in fresh medium. After that, the viability is measured, and this is possible. There is possibility of cell death due to storage stress, so the viability can be found at any stage, and it is calculated from the formula that number of cells which are growing divided by number of cells which are being thawed multiplied by 100. Then is the next step is the plant regeneration. The viable seeds are cultured on non-specific growth medium, and suitable environmental conditions are maintained, and plant cells are regenerated.